DNA represents the blueprint of the human genetic makeup. It exists in virtually every cell of the human body and differs in its sequence of nucleotides, which are molecules that make up DNA. We use the letters A, T, G, and C, or adenine, thymine, guanine, and cytosine, to represent this.
The human genome is made up of 3 billion nucleotides, which are 99.9% identical from one person to the next. The 0.1% variation can be used to distinguish one individual from another. It is this difference that can be used by forensic scientists to match up specimens of blood, tissue, or hair follicles to an individual with a high level of certainty.
The complete DNA of each individual is unique, with the exception of identical twins. A DNA fingerprint, therefore, is a DNA pattern that has a unique sequence such that it can be distinguished from the DNA patterns of other individuals. DNA fingerprinting is also called DNA typing. We are forensic scientists from Mohawk College, and our job is to obtain DNA from crime scenes and take it back to our lab to be analyzed to figure out who was at the crime scene.
The types of DNA that we look for are blood and any other bodily fluids, hair, skin cells, and any other types of DNA.
A group of us have just arrived at a crime scene. We're going to collect some DNA samples and take them back to our lab.
We're here trying to figure out who the murderer was and who the victim was, so we're going to try and find some examples of DNA. Some blood. So I'm collecting a sample of blood from the floor and the hammer, and I'm going to put it in this bag. Collected a sample of skin cells from the handle of the hammer, and now I'm looking for some hair. Hold on. So I found a sample of hair, which could be from the possible victim of the murder.
Now we're going to go back to our lab and analyze our samples.
Julia, what is DNA fingerprinting?
DNA fingerprinting is a method of identifying a sample of DNA and comparing it to another sample of DNA.
A restriction enzyme is added to the samples to be compared. The enzyme cuts the DNA into smaller fragments. The DNA fragments are placed on an electrophoretic gel. When an electric current is applied to the gel, the DNA fragments migrate through the gel toward the positively charged pole. The smaller the fragment, the faster it will migrate. After a set time, researchers identify fragments of different lengths by staining the gel or by specifically highlighting fragments that contain tandem repeats.
We have 2 possible suspects and 2 possible victims, and we're going to compare the DNA that we collected to their DNA and see which are matches.
So our DNA samples came back from the lab, and we have the DNA samples from 3 of the possible suspects and 1 of the samples that we took from the crime scene.
So what suspect do you think matches the sample we took from the crime scene?
It's suspect 2. Should have been the one that you picked because it matches, as you can see, all of the little dark and lights match up.
DNA fingerprinting can be used in paternity testing to find out who the father of a child is. Without this, fathers could leave their children and not have to pay the fees.
DNA fingerprinting can be inaccurate because of sample contamination or laboratory error. In one case, DNA indicated that a woman was not the mother of her children until other evidence showed that she was a chimera, which means that she had different DNA in different cells. Tess, what are the Canadian regulations for DNA fingerprinting again?
Well, Canadian police have been using DNA fingerprinting to solve crimes for almost a decade, and they find it a really helpful tool. Without it, we probably wouldn't be able to solve as many crimes as we do.